Uh, I'm going to start over. Hi, everyone. My name is Sarah. I am SNEV president, and I would like to welcome you all to the second edition of the SNEV virtual conference. Our first edition was in 2021, and the idea of creating a virtual conference came from the fact that we wanted to create something for EV students that would allow you to network, was free, you didn't have to travel, and you could still share your work and have great prizes. For those of you that are not familiar with SNEV, I'm going to do a brief introduction. SNEV was started by a group of students in June of 2020 in an effort to connect students all over the world that were working on the EV field. And it is the first international student network focused on EV. We wanted to provide a platform for students to discuss, network, collaborate worldwide, and we're very lucky to have ISEV support. And since last year, 2021, they have been helping us make the platform grow, and they were like very nicely to provide the prizes for this conference. This is the current SNEV ECM from 2022 to 2024. We have students from all over the world in the ECM, from North and South America, Europe, Asia, and Oceania, and SNEV has been growing since its start, and now we have students all over the world, and we have 903 members currently, and we're very close to 1,000, so we're very excited with that. This conference was organized by this group of amazing people. Carlos and Same are the conference managers, and I would like to thank everyone that was involved for their efforts and for the work they put together to bring the conference to you. Now I'm going to pass to Same, which is going to tell you a little bit more about what you can expect for the next 2 days of the conference.
Yes, thank you, Sarah. I think everyone should be able to hear me now. My name is Sam. I'm a MD, PhD at Wake Forest in North Carolina, one of the SNEV executive committee members, and for now the virtual conference co-conference manager. And I just wanted to say thank you to everyone who is attending the virtual conference today. We will have just several keynote speakers throughout the day. As you can see here, we have these individuals listed from, and what we're pretty proud of is that they're from all across the globe. Do we lose the PowerPoint, or am I just not seeing it? Oh, there it is. Yeah, so
sorry.
No, you're okay. They're from all across the globe. Here in the United States, we have Halal Rather at Duke University. We have individuals who are also from the United Kingdom, as well as from Europe, Belgium, and Netherlands. And we have representation from the oceanic state countries as well, such as Australia. Yeah, if you could go to the next slide, Sada. And this is the schedule that we have for our virtual conference. We'll have just live sessions during 2 days across different time zones. Our idea here was to make this both Europe and Asia friendly. And the second day is more friendly to the American and European time zones. These schedules should have been sent out to everyone. Yeah. From this, we'll have just kind of like going through the flow of what will happen. During each of the sessions, we'll have a poll that will be released. During this, you'll have a time in which you can answer these polls. There'll be breakout sessions in which you can enter breakout rooms, discuss some of those things that are going on. And then, Sarah, next slide, Sarah. And the last day, there will be prizes for oral and poster presentations, as well as a poster at the end of the session that we would like to thank for one of our main sponsors, ISEV. And ISEV is providing some of the prizes that we're giving out for the best poster, best oral presentation, and best poster presentation. Okay. And I do want to say that we do have an active Discord in which individuals can join, and they can have conversations with others currently in the SNEV session, and I can drop that here. So you should have access to this Discord. Feel free to hop on if you are a Discord user and have conversations with others throughout the entire virtual conference. On behalf of just the SNEV virtual committee, we welcome you, and we hope you guys enjoy the conference.
Thank you, Same. I'm going now pass to the Emily, Natalie, and Maimona. They are going to be the moderators for the next session with the keynote speakers. So Natalie, you'll go first if I'm not wrong, and you can introduce the first keynote speaker.
Yeah, perfect. Thank you, Sarah, and thank you, Sam. So yeah, welcome everyone to day one of the SNEV virtual conference. So our first speaker for today in this session is Antonia Reale. So Antonia, if you want to pop your video on so we can see you. Perfect, thank you. So I'll just do a quick introduction before I get you to share your screen and we check your audio. So Dr. Antonia Reale is a medical scientist at the Myeloma Research Group at the Australian Centre for Blood Diseases at the Central Clinical School in Melbourne, Australia. So she completed her medical degree in 2010 and training in medical oncology with honours in Bari, Italy in 2016. She was awarded an Australian Government Research Training Program and Monash Departmental Scholarship in 2018 in Melbourne. And her project involved the study of extracellular vesicles in multiple myeloma as potential drivers of disease progression and liquid biomarkers. So she has since completed her PhD in October 2022 and is currently validating her PhD findings. And she is now registered as a clinician in Australia, and she'll soon commence practice of medical oncology in Australia. So well done, Antonia, and thank you so much for joining us. So yep, we can see your screen. That looks great. Did you want to say a quick hello so we can just check your audio too?
Hi, everyone. Thanks for inviting me. I'm very honored to be here today to share my work, actually my journey. And thank you so much also for your incredible work. I know it's hard work, and just well done. Thank you. So thank you also for the introduction. So I won't need to say anything else to introduce myself. My work, and so this presentation will basically is based on multiple myeloma, which is blood cancer. And my EV work is based on understanding the roles of EVs in this blood cancer, multiple myeloma. I won't go into details because there are so many things to know and to tell about myeloma, but the main message here is that myeloma is a highly heterogeneous disease, a highly heterogeneous cancer. And unfortunately, despite advances in its treatment and advances in the knowledge of its biology, it remains incurable. In fact, we have several unmet needs. We need to better understand the significant heterogeneity of this disease. We need to improve diagnosis and monitoring modalities. We need to better risk stratify patients, and we need to find new targetable mechanisms and molecules. When I proposed my PhD project, we are talking about 2016. I had just completed my oncology training, and my professor asked me to come up with an idea, a new idea for my PhD. And my attention was captured by this new field to me, the EV field. And in myeloma, there wasn't very much out there in terms of publications, papers. I didn't know that there was going to be an explosion, not only in the EV field, but also in myeloma. And although this is a bit outdated now, it's I think I generated this in October last year. But as you can see, there has been an increasing number of published papers, and I am very happy and honored to have contributed to this. And so I was right. My professor supported me. He encouraged me to pursue this project. And here, I just wanted to explain you a bit more why EVs and how they would fit with our work as a group. So my group has been interested for several years in liquid biopsy. I'm not sure if you all know what liquid biopsy is, but basically is a method that enables the analysis of biofluids. In our case, we use mainly blood, but there are other biofluids that can be used. And by the analysis of the content of biofluids, you can understand how, for example, cancers behave. You can detect factors like proteins, DNA, RNA, EVs that can help predict, for example, the response to therapy. They can help stratify patients, can help diagnose patients. And so my group has been working for several years in the space of DNA and RNA as liquid biopsy, while I was proposing EVs as liquid biopsy and therapeutic target. And the reason is that the EVs have a particular cargo profile and biophysical properties that make them useful as liquid biomarkers. We know that they play an important role in cell-to-cell communication, in pre-metastatic niche formation. For people that don't know what this is or don't work in cancer, pre-metastatic niches are basically areas in our body, this can be in any organ, where cells of the microenvironment, or cells in these areas, are prepared by several mechanisms to accommodate the arrival and the growth of cancer cells. And EVs have been shown to play a role in this space as well. They have a role in cancer progression, and this is all supported by the recent introduction of EV-based liquid biopsy into clinical practice guidelines for prostate cancer. We, other groups, including our group, has confirmed that EVs have a role in cell-to-cell communication, and in particular in myeloma. Basically, myeloma cells grow in the bone marrow, in bones. That's the typical disease, is characterized by the outgrowth of these cancer cells in bones. You can have fractures, bone fractures, severe pain, among many other signs and symptoms. And so the hallmark of myeloma progression is the crosstalk between myeloma cells and the microenvironment. EVs participate in this crosstalk, and this has been proven also by other groups, as I mentioned. But we have several unmet needs. We need to standardize the methodology. We need to establish a role for EVs as biomarkers. We need to establish a targetable role for myeloma EVs as cell-to-cell communicators. And we need to establish a role as mediators of the formation of defined pre-metastatic niches. A few years ago, I did this comparison. I looked at how myeloma researchers isolate EVs compared to other EV researchers. And as you can see, there's a significant difference in the way myeloma researchers isolate EVs. Unfortunately, the method of choice is precipitation, which has we all know, or probably some of you will learn over time, is not the gold standard and is not ideal to isolate highly purified EVs. And so when I defined my project and when I started working on EVs, I decided to look at both cell culture supernatants and plasma, blood plasma from patients. And so this was my study model, and I looked at several pre-analytical variables We know that the volume of starting material is very important. We know that the samples, if fresh or frozen, can provide different amount and EVs with different characteristics. So I looked at that as well. I looked at cell culture conditions, and I looked at different blood collection tubes. This is all published. So my message here is that ideally I wanted to use one method for both supernatants and blood, but unfortunately this was not possible. And the reason is that basically for supernatants, I chose to use density graded centrifugation, which is recognized as one of the best methods to isolate highly purified EVs. But this requires specialized equipment, is time consuming, and also importantly requires high volumes or concentrations of starting material. Unfortunately, these characteristics are not compatible in a clinical setting. So I chose to test commercially available kits. This is because they are easy to use. They require less amounts of starting material, and the processing time can be very short. So I tested a few commercially available kits for blood plasma, and then I chose to proceed with all my experimentation with the Norgen kit. I just want to specify also that my work has been looking at both large EVs and small EVs, but then I focused more, for practical reasons, I proceeded with small EVs, and the presentation is more around small EVs. And so this is a model that I've been working on. Basically, I've been able to confirm that myeloma cells secrete EVs. I've been able to isolate EVs from supernatant, cell culture supernatants, but also from blood plasma. And importantly, to confirm the role of EVs in the crosstalk between myeloma cells and stromal cells, I've been able to confirm that stromal cells, which membranes are in red, nuclei in blue, are able to uptake small EVs derived from myeloma patients, which are the green dots here. You can see other cell types. These are premalignant conditions, and I have been using also EVs from these patients, and also healthy donors as controls. And so here you can see these are videos that we have generated in collaboration with Irena Carmichael from our center. And you can see how over time there's an increasing uptake of small EVs. And here, if you focus your attention on this green dot, you can see how the stromal cell membrane projection is capturing the EV. And I find this very fascinating. And so after confirming that stromal cells can uptake EVs from myeloma patients at a higher extent compared to healthy donors derived EVs, I proceeded at looking at how these EVs from myeloma patients can alter the function of stromal cells. And so I've looked at, after treating stromal cells with EVs from plasma of myeloma patients, I've looked at the proliferation of stromal cells, migration of stromal cells, and then I also looked at the adhesion between stromal cells pretreated with EVs with cancer cells, so myeloma cells. And I've observed an increase in proliferation, in migration and adhesion. Importantly, I've observed an increased migration and adhesion also when stromal cells are pretreated with EVs from the premalignant conditions, so indicating that EVs have a role not only in the progression of myeloma when it is an active disease and a role in drug resistance, but also a role in the progression from premalignant stages to an active disease, more advanced disease. And this is important because in myeloma, we don't treat patients with premalignant conditions, but over time, thanks to the increased knowledge on the biology and thanks to the way we are able now to stratify patients and to understand how patients that, although they have premalignant conditions, they can have specific characteristics that make them at high risk of progression And so today, in some instances and some centers are able to treat these patients, and they benefit from this treatment. And so EVs are now, I'm able to show that play a role in this space, and so they may contribute, hopefully in the future, in improving the treatment strategies for myeloma patients. And so, is this a new targetable approach? Obviously, it's a bit still early to answer this question. Although I completed the PhD, I progressed my work. I'm trying to finalize some experiments to be able to publish my work by the end of the year. But anyway, the message here is that after proving that stromal cells have an increased proliferation and migration after treatment with EVs from myeloma patients, there's an increase in addition between stromal cells and myeloma cells. But also, and importantly, there's an increase in proliferation of the tumor cells. And this is confirmed by two methodologies, by direct counting and flow cytometry. And there's also increased drug resistance. So by using an anti-myeloma drug that is typically used in the clinic, which is called bortezomib, BZ, I observe a reduced cell death, percentage of cell death of tumor cells when I pre-treat stromal cells with EVs from myeloma patients. This is very interesting, and it's been confirmed by other groups in not in the exact same context, but in a similar EV-based context. And so the next question was, what are the possible oncogenic drivers of all these functional changes? And so we've been working with Associate Professor David Greening, and we've been looking at phosphoproteomics. We've treated stromal cells with EVs derived from patients with myeloma, and after a very short time, we performed phosphoproteomics. We've been able to observe an increased expression of several proteins of interest. And importantly, there are a few kinases that are possible activators of these phosphoproteins. And even more importantly, all these proteins can be targeted, have been shown to play a role in other cancers, in myeloma, and can be targeted with new drugs. And so then we've looked at also gene expression of these stromal cells treated with EVs from myeloma patients compared to controls, healthy donors, and premalignant conditions. And even here, we are able to observe an overexpression or downregulation of genes. And importantly, some of these genes are involved in the activation of transcription factors that play a role in cancer progression and also in myeloma. I was captured by this, basically by doing gene ontology analysis. There is an enrichment of the epithelial to mesenchymal transformation pathway. And this fits very nicely with all my findings that I've described, and introduces also my next experiment, which I've completed recently. And this is a pilot in vivo study using an established myeloma model, where I have preconditioned mice for 3 weeks with EVs from myeloma patients. Then I've injected mice with cancer cells, and I've observed tumor growth for 6 weeks. The most important observation is that after the 6 weeks, after culling mice, I was able to observe an increased number of cancer cells in organs like kidneys, heart, liver, which in myeloma represent what is called extramedullary disease, so an atypical form of disease, which is typically more aggressive. And I observe, in fact, less presence of tumor cells in the bone marrow, which is the typical organ where myeloma cells grow. I didn't observe a significant difference in lungs and brain. This is very encouraging. It's only a pilot study, and now we are performing immunohistochemistry to confirm that EVs have prepared the niches, the pre-metastatic niches, to accommodate the arrival and growth of myeloma cells. One last concept that I would like to share with you is that we've been also looking at the content of EVs themselves, EVs derived from myeloma patients compared to healthy donors. We performed a preliminary analysis, mass spec analysis. This has been published, and interestingly, we've observed an upregulation of proteins associated with cell migration and cell adhesion. And also in this case, there's a nice correlation with all the functional findings that we've observed and described. And so we then expanded the number of samples. This will be part of the next work that hopefully will be published this year. Here basically you can see how the EV proteome basically reflects the heterogeneity of the disease. So we are not surprised by this, but healthy donors derived EVs cluster pretty well differently from the disease compartment. And we have been able to detect several proteins, almost 400, and these proteins, several proteins upregulated or downregulated, are able to discriminate disease from non-disease. Importantly, the proteome of EVs from myeloma is specific. This is by doing an analysis of our sample set, but also comparing our sample set with published sample set in myeloma. And importantly, there is not much overlap with proteomes from other cancers, so confirming that this proteome is specific for myeloma. And finally, we are also looking at the protein concentration of EVs. There will be also NTA, but we are finalizing the analysis. And basically, with our initial sample set, we weren't able to see a difference in the mean, but I was able to see a difference in the variance in the myeloma group compared to the controls. And so we've expanded the number of samples using a biobank, and now we are able to see a difference in the mean. This is not even updated. I have more samples and the data is confirmed, and also this has a predictive value by ROC analysis. But I will implement the analysis, complete the analysis, and hopefully I'll show you this at the next opportunity. And so basically, in conclusion, this strategy may provide important insights into targetable mechanisms of disease progression in myeloma, and the characterization proteomic profiling of disease-specific circulating small EVs as a biomarker discovery strategy may provide translation applications in myeloma. I would like to thank my group, and in particular my supervisors, Professor Andrew Spencer, Dr. Tiffany Kong, and also the RAs Mala and Victoria. And also I would like to particularly thank collaborators who have been very supportive of this work and have also guided me through this challenging journey, but very fascinating journey. Associate Professor David Greening and Dr. Ram Tsu. And again, I thank you for giving me the opportunity to share my work.
Thank you so much, Antonia, for that wonderful presentation. That was really exciting. And speaking as someone who's also done biomarker discovery work using EVs, I understand how challenging it must have been, but you have done an absolutely beautiful job there. So congratulations on your PhD completion and your continuing work. So I'd like to open up the floor now for any questions that anyone might have. For Antonia, so you can raise a hand or unmute yourself. And I can't see any hands raised just yet, so I can definitely get the conversation started. So you mentioned David Greening, and for those of you who don't know, Associate Professor Greening is the president of the Australia-New Zealand Society of Extracellular Vesicles, and definitely an emerging leader in the proteomics field. So you're in very good company there. And I wondered about the phosphoproteomics. That collaboration and getting that started with him, how did you go about that? Was that something that you approached him, or did you meet and have a discussion? Because obviously building these networks during and after your PhD are incredibly important for continuing work. Obviously collaboration breeds success in a lot of cases. So can you explain how that happened?
Yes, so that aspect of the work has arrived basically in the last 1.5 years of my PhD. after we had already performed mass spectrometry together on EVs. So the original idea was mine. I came up with the idea of performing this work in the myeloma context. But then I was lucky enough in life, I guess we also need to have to be lucky, that when I went back to work to start the PhD after maternity leave, a bioinformatician joined our group at Monash University, Alfred, and he had just completed his PhD with the group of Associate Professor Greening and Professor Richard Simpson in Melbourne. And so he basically started this collaboration, and I started communicating with Greening and also with Dr. Rongsu, who is now in my department at the Alfred Monash. And so basically they have this, although I had already designed the project, they obviously had guided me from the start because I didn't have the knowledge. I was new to the field. And so we've been having ongoing discussions since day one. And obviously, I think I was lucky because without their support, their help, their guidance, the project could have had a bad outcome. And I say this because initially I had chosen only to use commercially available kits, also for supernatants. And I did test these in supernatants. There's a lot of other things that I have done that I could not show you today. Just because of timing issues, we could have been here for hours describing all the things. But yeah, as we know, commercially available kits can be good, but can be also not good. I don't want to say bad. And so it's surely not good in my experience for supernatants. And so then we've redefined things over time. And as you know, he's one of the leaders internationally recognized in proteomics. And so we've performed mass spec. In regards to phosphoproteomics, so going back to your question, after performing all the functional experiments, after proving that EVs had an effect in the functions of stromal cells, I was curious to know what is driving this. And these are questions that I was asking by reading other researchers' work, by joining conferences and having discussions with other EV people, and then with David having him big knowledge in EVs and proteomics, and also expertise in phosphoproteomics. So he suggested to perform this experiment, and it's been a very nice experiment, which now will be validating. Obviously, all these experiments that you've seen have had all a preliminary phase, optimizing. So I did some experiments looking at some proteins like ERK and phospho-ERK. And so that's where we defined the timing, so when to perform the actual experiment. But I was interested in very, very early drivers, and that's why it was done within 5-10 minutes. Some other groups performed this experiment at half an hour or 1 hour as well. But I was interested in the very early changes. And then that's why RNA sequencing was performed afterwards, because then I was hoping to find some connection in activation of phosphoprotein and then genes that are involved in cancer transformation. And the story is fitting very nicely. So I'm very excited. I'm actually preparing the manuscript. So I didn't want to show you all the genes, all the proteins, just because I'm preparing the manuscript, but I was happy to show you all my journey.
Yeah, of course. No, obviously, yeah, again, biomarker projects, it's a very long timeline because you do all the work, you get all the exciting results, but then you also have to then go and validate everything. So I completely understand. We do have a question in the chat box from Sebastian. So if you would like to unmute yourself to ask your question. Otherwise, I'm happy to just read it out from the chat box. So he says, have you conducted research on plasma cell-derived EVs specifically or on EVs in general?
Sorry, can you repeat the question?
Have you conducted research on plasma cell-derived EVs specifically or on EVs in general?
Oh, so no, plasma cell EVs using has controls the pre-malignant conditions and healthy donors, so no in other conditions. But going back again to your question, this journey, and by participating in conferences and also in our internal seminars, my work has been exposed to other researchers, and recently I have applied for a small grant, but in collaboration as a co-PI with another ECR, and we have received funding to start a project on epilepsy, so another context, so looking at the role of EVs in epilepsy. So now, and also I've had discussions with other colleagues in other cancers. So I guess after completing the PhD, after publishing a few papers, after having so many conversations with a lot of researchers in the EV field and other fields, obviously, I guess, like right now, I'm in Italy and I'm working in my old department for a few months before I start my new job in Melbourne in August. I have obviously a lot of, I know a lot of people, colleagues, oncologists, and so these conversations are easy to have. But having said that, I knew no one in the EV field. And by joining, by collaborating with Associate Professor Greening, by collaborating with Dr. Rong Su, by participating as an in the ECR subcommittee for the Australian New Zealand Society, I've been exposed to a lot of EV people, and we've had so many conversations. This is how then things progress, how new ideas can come. And obviously, if you have the support of your supervisor in this, because I understand not many leaders support this during the early years of our career. So if you have, but I understand in the EV field, I guess everyone is supportive of this. So we are very lucky to have leaders that support this networking, and being part of societies like SNEV has been a great, amazing opportunity as well for all of you. But for me, I joined from the start, and I know a few of you. Yeah, that's how I think networking, participating, being part of committees is the key to implement your work, and also to explore other fields like this is happening for me.
Yeah, that's a very interesting transition from myeloma to epilepsy. So things are opening up for you. That's wonderful. So Maimona, did you want to ask your question? Did you want to unmute yourself?
Yeah, so yeah, I would like, thank you so much for the interesting talk, and yeah, the inspiration you're giving us as early researchers as well, doing PhD in a place that has not done EV research before, and yeah, emphasizing on the importance of having these collaborations and attending conferences and sharing ideas with others. So my question is about, how did you track your EVs in vivo? What method did you use? Yeah,
I still haven't done that. It's in the. We planned it, but as you know and understand, it's very difficult to do everything. You cannot do everything, and surely you cannot do everything at the same time. So all that I've shown has been done since 2018. And obviously over time we've planned a lot of other new things, and that is part of our future work. So we have not tracked EVs in vivo. What I've done is I've treated mice with EVs, and then I've collected organs at the end of the experiment, and I've analyzed the presence of cancer cells in different organs, but I have not looked at EVs themselves.
okay. Okay, thanks. Thank you.
And I think we still have time for another question or two, if anyone else wants to put something in the chat box. But I also wondered, so for your controls, did you do like a healthy control EVs effect versus the myeloma EVs, and if there were similar changes or no change, or you know, there's obviously like a million different controls you could do, but I just wondered if you did a healthy donor control EV in your experiments. I think I did see something, but I wasn't sure if it was
in all the experiments, you will see ND, which is non-disease, or HD, healthy donor. So so we are lucky enough to have access to samples, both patients and healthy donors. This is another key for research, you know, if you are able to access samples, but if you are not, that's where collaboration comes in. You reach out to centers. And for example, now, you know, we can easily access samples. So feel free to reach out. There's always space for chats and collaboration. So all my experiments have controls. Healthy donors, in some instances, don't have very much an effect, but this could also be related to the specific experiments that I've done. There are all sort of other functional experiments that can be performed, and I'm sure they have an effect. We all know by literature that EVs have a role also in healthy conditions. But when you compare, in my case, with cancer, myeloma EVs, there's a significant difference in the effects. And so, yeah, you will see all healthy donors is everywhere, basically. And also, I'm lucky enough also to use the premalignant conditions, which are conditions that typically are not treated, as I mentioned before. But the reason why I use them is because we can access them. But importantly, I believe that increasing the knowledge also in that space, by increasing the knowledge in that space, patients will benefit. Maybe I won't see it probably in the next 10, 20 years, but someone else will. So I believe that's very important. So when you choose the controls, obviously you need to have an understanding of the disease. And if you have access to controls, not only healthy donors, that's very important.
Absolutely. Yeah, because EVs are magical things, and I guess we want to make sure that it's not all EVs that are doing these things, but specifically the ones from the condition that you're interested in. So,
and also another, I guess, sorry, just an important concept
it's fine.
Is obviously with the commercial kit that I've used, EVs are not highly purified. They are enriched. And for people that are new to the field, this is an important concept. Obviously, in these samples, there are other factors, core factors, confounding factors. There's different names, but basically it's not just EVs. So another important control, which I have in some experiments, not all of them though, at this stage, is to use the buffer or the whatever material, biofluid, after you enrich EVs. So whatever is left over, that is a proper control as well, because that will contain factors that can confound findings. So obviously, ideally, you want to isolate highly purified EVs, but this is not always possible. And especially when you want to apply your knowledge in the clinic, you also have to consider how the method, how is that performed, the timing, the cost. So you cannot do everything in a clinical space. So you always want to find a middle way where you can do a proper research, but also that you can apply it in a clinical setting.
Yeah, many things to consider. An experimental design is absolutely key. And yeah, I think you've really nailed that down in your work. So I think at this point, there's no more questions in the chat box. And I think our time is just about up. So I'd just like to say thank you once again for speaking today, first speaker of the conference, and very best of luck with your ongoing work, Annie. Thank you very much.
Thank you. Good luck to you and thanks for your work.
Thank you.